Another kind of disorder we can have are dominant disorder, autosomal dominant disorders. Now, in a dominant disorder, you only need one copy of the affected allele in order to end up with that condition. After all, that's what dominant means. Achondroplasia is the most common form of congenital dwarfism that exists, and it is in fact a dominant trait. So, if two people with achondroplasia go to a genetic counselor and they say, we want to have some kids, what are the odds that we are going to have offspring that also have this condition? Well, the genetic counselor will say, okay, Mr. and Mrs. Smith, you are both heterozygous for this condition. The reason why will be obvious in a moment. There would be a 25% chance, remember, this is a dominant disorder, so there's a 25% chance that two of these achondroplasia genes will be passed on to your offspring. There's a 50% chance that you will have a heterozygous child, but with achondroplasia. And there's a 25% chance that you will have a child with two recessive genes, which in this case means a child of average height. You would never know that their parents have any sort of genetic condition just from looking at them, because they look the same as the majority of people. Now, you would normally think, okay, that means that we must have a 3-to-1 phenotypic ratio, because we have three situations with this dominant allele and one without it, right? But that's actually not how it works with a lot of dominant disorders. As it turns out, if you are homozygous dominant for achondroplasia, that is a lethal combination. You do not see that zygote actually come to term. It never actually survives long enough. So everyone with this condition turns out to be a heterozygous individual, which means we don't get a 3-to-1 ratio in the offspring. We actually see instead a 2-to-1 ratio in the offspring. 2-to-1 means that there is a 66.6 repeating percent chance of someone being born with this form of dwarfism, but there's also a 33.3% possibility of being born with average height. So about 1 in 3 odds of an average heighted child being born from these two parents. Now, I think this is maybe one of the best examples I can give you for the difference between a dominant trait and it being the most common trait in the population. Achondroplasia is a dominant trait. If you have even one copy of it, you are born a dwarf. But it's clearly not the most common trait in our population, right? So whether or not something is common has nothing to do with whether or not it's dominant. All right. Another chromosome disorder, another genetic disorder, is called Marfan syndrome. This gentleman's name is Niccolo Paganini. Here's a little bit of the violin music that he plays. At the time, he was considered to be one of the greatest violinists in the world because of these really complicated types of performances he could give. His fingers were abnormally long and dexterous, and extremely strong as well. And he could actually pluck the strings of the guitar with one hand while he was simultaneously playing it with the the bow in the other hand, which meant he could do things that other violinists simply weren't able to. And we're pretty sure that Paganini suffered from what's called Marfan syndrome, which happens in about 1 in every 5,000 live births. This is a dominant disorder, so he only needed one copy of this allele in order to end up with this disease. We actually think that Abraham Lincoln may have also had this disease. So certain things that are associated with it: long, lanky limbs, the connective tissue in your ligaments, your collagen is particularly elastic, and that gives increased joint mobility and flexibility. However, over the long term, it puts a lot of strain on those ligaments and tendons and that connective tissue, and you start to lose a lot of mobility later in life. So he's suddenly had to start canceling a lot of performances. But in his heyday, people would tell stories and say that he had sold his soul to the devil for his incredible talent with the violin, because no mere mortal could possibly do the kinds of things that he was doing with the violin, and he absolutely played into that. He thought that was really good branding for him. This is an example. Marfan syndrome is not a disease that anyone would really want to have, but in some situations you might find that even if you have this disorder, there might be some advantages that it ends up conveying to you. He did very, very well for himself financially as a violin player, partially probably because he had this disorder, even though it kind of shortened the total amount of time he was able to play for, because eventually he just couldn't keep on these performances. Not every gene in every organism follows Mendels rules precisely. Mendels rules are a really good way to understand the basic mechanisms of genetics, but they don't 100% hold true every single time. So for example, we've been using pea flowers and pea flower color as a big example up until now, but let's take a look at a different kind of plant, a snapdragon. Snapdragons have red flowers or they have white flowers, just like peas have red flowers and white flowers. You can create true breeding varieties just the same way Mendel did with the peas. However, when you crossbreed, when you get hybrids in the F1 generation of snapdragons, you actually end up with pink flowers. So the hybrid, the heterozygote, does not look like either of the two parents. You actually see a blending of the two phenotypes here. We refer to this as incomplete dominance. This is where the hybrids have an appearance which is halfway in between the phenotypes of the two parents. So red plus white gets you pink. Another example might be someone with straight hair and someone with curly hair. They have kids. Their child has wavy hair. The wavy hair is about halfway in between the phenotypes of curly and straight hair. Now, when you do a F2 generation, when you cross two of these pink hybrids with each other, you end up with the same genotypic ratio that you would have before, because that really hasn't changed. You still have 25% odds of getting a homozygous dominant individual, 50% odds of getting a heterozygous individual, and 25% odds of getting a homozygous recessive individual. So your genotypic ratio is still 1 to 2 to 1, just like in Mendel's experiments. However, if you look here, every genotype now has a special phenotype associated with it. So now, instead of a 3 to 1 phenotypic ratio, you end up with a 1 to 2 to 1 phenotypic ratio as well. 1 red snapdragon for every 2 pink snapdragons for every 1 white snapdragon. So that's incomplete dominance. In humans, we have a form of incomplete dominance in our genes for receptors that take cholesterol back out of our bloodstream. So you might have a condition which is called hypercholesterolemia, which means that you do not pull as much of the low-density lipoproteins, the bad cholesterol, out of your bloodstream as you ordinarily should. Now, if you have two dominant alleles for the gene that makes these receptors, you are going to have normal blood cholesterol. You're going to pull plenty of bad cholesterol out of your bloodstream. If you have homozygous recessive genotype for this particular gene, then you're not going to make any of these receptors, and you're going to have blood cholesterol level that's 5 times higher than a normal person's. However, if you are heterozygous for this trait, if you have one dominant and one recessive allele, then you'll make about half as many receptors as a person with 2 dominant versions of this gene. So you'll end up with about twice as much blood cholesterol as the average person would. So you can see here that we have 3 different states. The homozygous dominant individual makes lots of these receptors. The heterozygous individual makes some of these receptors, and the homozygous recessive individual makes none of these receptors. So you have 3 different phenotypes depending on what genotype you have. That's incomplete dominance. The heterozygote condition is halfway in between the other 2 extremes. Okay. So, I mentioned blood type a little bit earlier on. We talked about positive and negative blood type, but that's not the most famous kind of blood type. I think most people know the ABO blood group, and that's a little bit more complicated than the kinds of genetics we've been dealing with so far. So what are your blood types? That should probably be the first thing that we explain here. On the outside of your red blood cells, intercalated in with the cell membrane, you have little carbohydrates, which essentially function as a kind of name tag for your red blood cells. It's an identification system so that your immune system can recognize self from non-cells. It needs to be able to identify, oh, this is Professor Klein's red blood cell here. This one's fine. Oh no, this is some foreign blood here. We need to get rid of that. It's basically to make sure that it can identify foreign entities that are maybe invading your system. You can have, for example, the A-type carbohydrate on your red blood cells. If you have the A-type carbohydrate, you have A-type blood. You could also have the B-type carbohydrate on the outside of your red blood cells. If you have that, you have B-type blood. But it's also possible that you have both A and B on the outside of your red blood cells in about equal proportions, and then you would have AB-type blood. It's also possible that you have alleles which don't code for some of these carbohydrates, in which case you don't make any of these antigens on the outside of your red blood cells, and then we would say that you have O-type blood. O as in you make 0 of these carbohydrate antigens. Now, your body is going to create antibodies against whatever kind of antigen you do not possess. If you have A-type blood, you will make antibodies in your bloodstream against B-type blood. So if any B-type blood gets into your system, your immune system will attack that. If you have B-type blood, you will make antibodies against A-type blood, and your immune system will attack that. When these antibodies attack these blood cells, they cause them to clump up with each other. They attach to the red blood cells in multiple places, and you get clotting, clumping, starting to occur. If you have AB-type blood, then you are fine with any kind of blood entering your system. You have both the A antigen and the B antigen, so your body doesn't recognize either of those as being foreign. If you have O-type blood, then all of these antigens appear foreign to you. A-type, B-type, and if you find some blood that is AB-type, that's also foreign to you. Now, as you might imagine, the genetics here are a little bit more complicated than we've been looking at. So the ABO blood types, we don't have 2 different kinds of alleles, we actually have 3 different kinds of alleles. 2 of them are dominant, and 1 of them is recessive. This should actually probably be a lowercase I. 1 of them is recessive. Now, since we have two different dominant alleles, we say that they are co-dominant. And in fact, if you have both of these alleles, you express both kinds of blood group. You have both kinds of antigens. So they don't just dominate, they co-dominate in this case. Let's take a look at this in a little bit more detail. Okay, so there's three different kinds of alleles you can have. If you have two copies of the recessive allele, that doesn't code for any of these blood antigens at all. So your red blood cells will have no antigens on the surface. As a result, your body will recognize any blood with antigens on it as foreign blood, whether it's A-type blood, B-type blood, or AB-type blood. You cannot receive any blood from a donor that is not O-type blood if you have O-type blood. However, because your blood cells do not make any of these antigens, you can donate your blood to absolutely anyone. No one's immune system will recognize your blood as foreign because it lacks the surface antigens to indicate that that is a foreign bit of material. Okay? As a result, O-type blood is referred to as the universal donor. Anyone who ends up with the dominant A allele is going to make the A-type antigen. That's what these carbohydrates on the red blood cell surface is called. They're called antigens, identifying molecules on the cells. Now that's true whether you have capital IA or two copies of capital IA. It's also true if you have only one copy of IA and a recessive I as well. In both these cases, you make the A-type antigen alone. If that's true, you will make antibodies in your body against the B-type antigen because you would consider that to be a foreign substance. Any kind of blood that has that B-type antigen, that is B-type blood or AB-type blood, it's going to clot up if it gets into your system. Your immune system will attack it with your anti-B antibodies. Similarly, whether you are homozygous dominant for the capital IB allele or you're heterozygous capital IB recessive I, you'll make the B-type antigen, and therefore you will consider A-type blood to be a foreign substance, and any blood that has the A-type antibody in it is going to appear as foreign, and it will clump up. Your immune system will attack it. So that goes for A-type blood and a B-type blood. If you have B-type blood, you can't have either of these two transfused into you and survive. Now, if this is the situation where it's different. Up here, everything's the same as what we talked about before, except there are two different dominant alleles. But what happens if you have both dominant alleles? Well, in this case, they don't dominate over each other. Both of them get expressed 100%. So you will make both the A antigen and the B antigen. And we just call that AB type blood. Now notice the difference between this and incomplete dominance. In incomplete dominance, you saw a blending of the traits in heterozygotes. Red flowers and white flowers give you pink flowers. But in codominance, we have 100% the A-type trait and 100% the B-type trait. You exhibit both of these phenotypes at the same time. Because people with AB-type blood consider both the A and B antigen to be familiar substances, to be their own body parts, it doesn't matter what kind of blood they receive, whether it has the A antigen or B antigen. So every kind of blood that they receive, it's not going to clot up. AB-type blood individuals are the universal receivers. They can accept anyone's blood, ignoring for a moment the Rh factor positive-negative thing, because that is a concern as well.